There are certain criteria a person must fulfill to pursue a successful career in STEM, which stands for science, technology, engineering, and math and medicine. However, these criteria should not be related to the gender, race, or any cultural background of this person. Instead, in my opinion, the three main criteria are, first, to be curious, to be persistent, especially when it's getting tough, and to be very passionate about your work. And if someone would ask me to define myself by just using 3 words, I would use exactly these 3 words. I'm Jennifer Zenker. I'm a research group leader at the Australian Regenerative Medicine Institute at Monash University in Melbourne. I'm Jenny Zenker. I'm a high-performance triathlete who trains twice per day to compete at the international level, including World Championships. I'm Jenny. I'm a swim and triathlon coach with over 8 years of experience coaching little kids to adults and also beginners from very advanced to high competitive athletes. As an athlete, I'm experimenting with the own limits of my own body, basically on a daily basis during training, but especially during races. So as you can see here in these pictures, especially in the picture in the middle where I'm running, I'm in pain. I'm hurting myself. All I want there at that moment is to stop and not running anymore. But I do continue because it's really incredible to experience what the human body is capable of, what the human body can achieve. And most of us are actually even not aware of what the human body is a incredible machine. So I, as a scientist, I want to understand how is that possible? An incredible machine, our human body, how is it formed from the very beginning on? What are we made of actually? So maybe have a look at yourself. What do you see? Well, I, as a scientist, basically see just a clump of 30 trillion cells. That's what we are actually made of. Every organ, every tissue we are made of is made up of cells, the very basic building block of our own body. And this system, our body, becomes even more fascinating when you start to understand that all this starts actually from a single cell, the fertilized egg. So this cell is no larger than 70 microns or 0.007 centimeters. And this single cell could have been you, could have been me at the very beginning of our lives. This single cell then starts to proliferate to become 2 cells, 4 cells, 8 cells, 16 cells. But if we would continue like this with just pure cell proliferation, we would probably nothing else than a mega massive mulberry and nothing else. So something else must happen during the early embryogenesis in order to allow us to become what we are, really a complex system of 30 trillion cells, which are very specialized and arranged and organized in a very high and difficult order. So one key process that happens now here at the 16 cell stage, so when the embryo is composed of 16 cells, is that cells start to become different, that it is possible to distinguish between 2 different types of cells. So here, the 16 cell stage, we can distinguish for the very first time inner cells, which are the pluripotent cells. So pluripotency means that these cells are able to give rise to all organs we are made of. Let it be the brain, the heart, the liver, skin, and so on and so on. As compared to the outer cells, which are determined at this stage to become the extra embryonic tissue. So, for example, the placenta and the yolk sac. And this process of cell differentiation basically occurs over and over again during our development to reach the final stage of our cells, for example, skin cell, heart cell, and so on and so on. So it's very important to understand these very fundamental processes that are occurring during the very first days of our development in order to understand how we develop, how we function, but especially also when a tissue or an organ gets injured or thick, to be able to repair it. Similar when a car breaks down, only the engineers that do understand how the car is assembled at the very beginning is also able to repair it. And that's the same basically with our human body. So in order to understand what's happening with the cells during the processes of cell differentiation, we need to be able to look into the inside of the cell and understand what's happening really on the subcellular level. So what's inside the cell? So inside the cell, you have kind of mini organs called organelles. The best known one is the nucleus, shown here as a first organelle. In the nucleus, you have all the genetic material, the DNA, and a lot of work has been done over the last decades to really advance our understanding how changes in gene expression really supports and triggers the process of cell differentiation. However, in the cell, there are many more organelles than only the nucleus. For example, mitochondria that are important for the energy production in our cells, and also many other organelles you can see here, but I won't go into further detail into each of these organelles. The important thing here is these organelles are basically in a liquid-filled environment and therefore can move around freely. However, this mobility of these organelles should not occur randomly, as I demonstrate here now in my slide. These motions and movements of the organelles must be highly regulated and also in accordance to each organelle so that they can interact with each other. And to allow this higher regulative mechanisms of the movement of the organelles to allow the cell to function and also to adapt to new requirements, the cell has a kind of a roadmap inside in order to guide the transport and the movement of these organelles. And this roadmap is composed of filament-like structures of the cytoskeleton system, as you can see here. So these filaments serve as roads to really trigger the directed transport of these organelles to certain regions where they are needed, and also the release or uptake of signals from the outside of the cell. So the cytoskeleton is kind of the framework inside the cell that regulates really the big majority of events that are happening there. So as cells then continuously differentiate into specialized cells, these microtubule and the cytoskeleton actually changes their conformation. So even these filaments-like structures are very dynamic. In less specialized cells, as you can see here, these filament-like structures usually grow from the center of the cell towards the periphery, forming this radial architecture. But during the process of differentiation, this filament cytoskeleton changes its architecture in a very specific way depending on the final differentiation of the cell, as shown here, for example, in the skin cell, where the anti-radial filaments reorganize into parallel filaments and therefore really allow the cell to change its shape, but also to reorganize the entire organelle architecture inside the cell as it is needed in order for a skin cell to function as a skin cell. The same process will occur in a muscle cell. The entire organization will be adapted so that the muscle cell can act as a muscle cell. So back to the cells in the early embryo. Here you can see a living mouse embryo using live imaging developing from the 8th to the 16th cell stage. And these are live imaging data that are also obtained in a similar way in vitro fertilization clinics in order to control the embryo quality used for in vitro fertilization. So in these kind of imaging data, you clearly see the separate single cells. You see when they divide and how they might change their position. But we are not able to see really what's happening inside of the cell with the arrangement of the organelles and the framework behind it. So really, to put light into this unknown area, we use a specific technology which allows us basically to color any kind of structure we are interested in here. So here you see basically the same embryo, but now the filament-like structures, the cytoskeleton is labeled in orange, the nucleus and the membrane in cyan, and now you can see when a cell divides how the cytoskeleton changes its shape, forming the spindle to allow the separation of one cell into two, how the membrane is really reshaping during these processes, and many, many more details we are now able to analyze. So by applying this technology, by really kind of color coding any structure of interest in the living mouse embryo, I was able to identify a new organization of these filament-like structures inside the cell of the embryos. Basically, inside of the cell of the embryo, you have a very unique roadmap as in no other cells seen so far. So basically, in these cells, you have a very dense bridge which connects two cells, which is a kind of bundle of a lot of filaments. And under the microscope, as the embryo is still living, I'm able then to zoom in into regions of interest in order to understand in more detail what's happening here. So zooming in into this very dense bundle, I can see that the growing tips, so just the tips of the filaments, are actually anchored at this point and growing away from there, growing towards the inside of the cell. So from there, all the filament-like structures start to grow from and then are redistributed into the inside of the cell, creating a very specific roadmap in the cell. So this was step one we needed to understand, to really understand the real-time processes inside the cell. Knowing now the architecture of this framework inside the cell, as a second step, we want to understand now why. Why do these cells need this very specific organization? And therefore, we are now combining our live imaging of the early mouse embryo with the cytoskeleton here shown in cyan, and then specific organelles we are very interested in. Here you see an example where all the mitochondria, so the energy-producing organelles, are labeled in orange. And again, by zooming in into regions of very high interest, for example, again, this bundle-like structure, which is probably the major highway in these cells of the embryo, you can see on the right-hand side a movie of this area, and then how exactly at this area really a lot of mitochondria are actually aligning along and getting really transported along these filaments with a major accumulation here really in the center part. So we still do not understand what does it mean. Is it a kind of exchange platform and so on? And further details we need to analyze further, really seeing that mitochondria that are transported at the moment are much smaller as compared to more static mitochondria further away from this kind of highway. However, we can, as the embryo is still alive when we are imaging, really look at very different and precise processes that are occurring during the early embryogenesis. As I mentioned, one key process is obviously cell proliferation, and cells divide, and one cell becomes two cells. So here's an example showing exactly the moment when an embryonic cell is dividing. In the zoom-in movie, again here on the right-hand side, you can see the spindle in cyan, so the filament-like structures that are really the guide of cell differentiation. And interesting is really then to see during the process of cell differentiation how these little organelles are pulled together with these cytoskeleton filaments into one cell versus the other cell. And this really allows us to understand much better how these different mitochondria are then divided into the two sister cells and how this might be different during various embryonic stages, especially when you have the segregation of inner versus outer cells. But also in areas just kind of randomly in the cytoplasm where we can't observe immediately a specific process like cell division or so going on, where it looks rather like a random distribution of mitochondria, as you can see here in the bottom right frame, where you see some distributed mitochondria again. We still do not understand if here the number of mitochondria, the distribution and the dynamics of the mitochondria are of any importance, and especially what's the pattern behind, what is needed in the dynamic and the organization of mitochondria to keep the embryo healthy, happy, and full of energy. And this is again a movie I will start playing in a second. It's a high temporal resolution movie. So basically, we image this region every single second for about 20 minutes. And this will give us a very good view what's really happening in real life in the early mouse embryo. And please pay attention to the bottom right corner when I start playing the movie. You will see immediately a very fast mitochondria basically shooting with a massive speed up here, but immediately stopping here when it reaches this other mitochondria. So I start playing it, and then you see shooting this mitochondria with high speed, but stopping immediately when it reaches this mitochondria, and then start somehow interacting or fusing with that. You see much more mitochondria here moving around. The movie will play now again, where I then trace the mitochondria with a gray tail, where you can see much better the movements of all these mitochondria. And this occurs so quickly, which was not possible at all to really realize or even to detect without the technologies we are using by color coding the structures of interest and using the live imaging technology we are doing. So basically comparing the situation of organelles versus our own organs in the body, it would be basically the similar situation that at this stage we would only have the knowledge that yes, we have a heart, yes, we have a gut, we have a lung, but we would not even understand that all these organs are interact with each other, that the heart really relies on the oxygen input of the lung, and the digestive system needs the support of the blood flow of the heart and so on. So all these dynamic processes, we have no knowledge about them yet on the subcellular level, looking at the organelles. So the aim of my research is really to create a motion map of the inner structure organization of the pluripotent stem cells. So the very important cells that are still able to give rise to all kinds of tissues we are made of. And therefore, I'm using the living mouse embryo. This will lead to fundamental discoveries of the first stages of embryogenesis. And this is a critical time period where, for example, in vitro fertilization processes or also in natural pregnancies, most of them might be actually lost. So why? So this is still a big question. And also, how is it maybe possible to detect or prevent the loss of a lot of pregnancies if it's non-invasive methods? So I'm not taking any sample out of the embryos or try to change anything with gene expression. All I do, as you can see here in the picture on the right-hand side, placing the living mouse embryo in a dish and then looking at it under the microscope by putting a light on it. And this will also allow us in the long term maybe to understand better how organs are actually formed from scratch. So as you can imagine, to get to this point was quite a long journey, and a lot of people really were very supportive and helpful on this journey. And it's pretty much impossible to mention every person who supported me on this way. But definitely there's some very key people that need to be mentioned. And it actually started all my entire biology scientist journey during school, where the biology teacher of my brother, who's 3 years older, asked him to do an experiment at home and gave him a couple of jars with Drosophila, so the fruit fly, so with some yeast in there. And he was supposed just to keep crossing them and to see if he can ever find some fruit flies with white eyes. So it's a typical Mendelian experiment. First, my mother was obviously not very enthusiastic about having a few hundred fruit flies at home, but when she realized how hooked both of us were, my brother and me, it was just amazing. And it was really the start of really my interest in biology. And since then, I knew I want to become a scientist. So I then, after also taking biology classes at with the same teacher, I studied biology still at home in Stuttgart in Germany. I continued then my PhD at the University of Lausanne in Switzerland with a really great PhD supervisor, Professor Roman Krust. I continued then my postdoc time at the Agency for Science, Technology and Research, ASTAR, in Singapore. And then I got the opportunity, or offered the opportunity of my life, to become a research group leader, offered by Pete Curie, now obviously at the Australian Regenerative Medicine Institute here at Monash University. Still, the trust he showed me by offering this position is just surreal for me, that I'm really now a research group leader. But this is also the stage in my career where my own passion, my own work won't be any more sufficient really to further succeed in future. I mean, it's still the key component, but really the foundation of that is really built up by my team members. And there I'm very thankful to have really like-minded, really enthusiastic scientists in my team. And I really appreciate all their work, which is really great and gives me really a lot of support. But also science is a very special field, a very special profession, because we really rely on external financial support. So all our science is based really on fundings, external fundings, fellowships, and scholarships. And therefore, I'm very thankful for all the support I got from the various fellowships and scholarships during my career. First of all, the German and the Swiss Science Foundation, and the Human Frontier Science Program, and also from CIFAR, a scholarship I received recently, so the Canadian Institute for Advanced Research. However, also for us scientists, it's a big responsibility to really make sure that the public, so the general people, start to understand what we are really doing, so that we come out of the dark and into the light, and that people start to understand the work we do is supposed to, in order to help everyone there, to make their life easier, to remain healthy and so on. So the advantage for everyone outside there, that's what our work is really based on. So the chance to give this seminar here today, offered to me by the Convergent Science Network, is a really great opportunity, and I'm very thankful for this, and I hope it really helps that really everyone, not only scientists, but also the public, start to understand what science is really about. Thank you very much.